This is chapter 21 that deals with the lymphatic and immune system. So the lymphatic system, it's composed of vessels similar to blood vessels, but not quite the same. These lymphatic vessels are throughout the body. There are some organs that are associated with the lymphatic system and cells that will carry and return any excess fluid to the blood. And before that extra fluid, which is called lymph, is actually returned to the blood, it's going to be filtered. And then the immune system, which goes hand in hand with the lymphatic system, is composed of cells and organs whose purpose is to destroy or neutralize any pathogens to prevent any type of adverse effects. It's basically your defense or your protection system. And so in this diagram, we can see that as I'm trying to get my pointer on, there we go. Kind of this greenish color here. These are all the lymphatic vessels that I just mentioned. So you can see they are throughout the body. So they can take any excess fluid and bring it back. It will be returned to the circulatory system up here. We'll talk about the points where it's going to come in. There are areas around the groin, the armpits, and in the neck where you have lymph nodes. We'll be discussing these as well, and that's where part of the filtration occurs. So that lymphatic system is associated with the circulatory system. That's where you see multiple systems intertwined and how they're dependent upon each other. If you remember in the circulatory system, in the capillary bed, where all that exchange occurs, as the blood is flowing initially into the capillary bed, remember you have the filtration, the substances flowing out of the blood into the interstitial fluid. So how much fluid are you talking about? Roughly 20 liters. That's quite a bit per day. That's per day. So how much of that 20 liters that filters out into the interstitial space? Remember, by the end of the capillary bed, you have reabsorption. So you've got to get the fluid back into the circulatory system, back into the blood. Well, only about 17 liters is reabsorbed. So that means if you're filtering 20, but only 17 is reabsorbed back, you've got those remaining 3 liters. That's quite a bit of fluid that has to be picked up. Notice throughout the body, it has to be picked up by the lymphatic system. How do you return those extra 3 liters back into the blood? Because if you don't, you're going to have a continual loss of volume from blood. You want the blood volume to remain fairly constant. So you got to get that fluid back in. Otherwise, you're going to be really uncomfortable because there's going to be a lot of swelling. How do you deal with this? So you have to get that remaining 3 liters back into the blood. Well, before you return it back to the blood, you're going to want to filter it, clean it up. You don't want to take a chance of having any type of foreign debris in there, like bacteria, viruses. Once, say, a bacteria gets into the circulatory system, once it gets into the blood, it can go anywhere in the body then. And you don't want that because that's going to make it a whole lot harder to treat. So you want to filter it. If you're picking up this excess fluid, this interstitial fluid, you may have had an injury site. You might have some pathogens, disease-causing microorganisms that are in that fluid. Well, you want to get them out before you get that in the blood. Now, lymph, if you're wondering, okay, so you have interstitial fluid and lymph. What's the difference between them? Lymph is when it's in the lymphatic vessels. I know it may sound kind of crazy, but yes, we give it a new name. So basically, it's interstitial fluid. When it's in the spaces between the cells, you absorb that into the lymphatic vessels. Now we call it lymph. Very similar to plasma. Then that's, in the circulatory system. Now, the lymphatic system, in terms of the structure of it, your circulatory system is a closed system, right? flows out of the heart, blood flows around the body, and returns back to the heart. It's closed. The lymphatic system is an open system, and it's a one-way system. So you start off with these very small lymphatic capillaries, and then the diameter of them will get increasingly larger and larger and larger as you get closer back to where you're going to return it into the blood. But it's a one-way system. The fluid is not pumped. Movement through the lymphatic vessel is going to be assisted by different movements of the body. It's going to, like contraction of skeletal muscles, that's going to help push the fluid up. Breathing, the changing in the pressures, that's going to help move the fluid up. The lymphatic vessels do have valves, very similar to the veins, that prevent backflow from occurring. There's 3 areas of the body, as I mentioned just a bit ago, where you have a higher cluster of lymph nodes. Now, you have lymph nodes throughout, but this is where you have a cluster of them. And that's in the inguinal or groin area, the axillary, remember it's the armpit area, and the cervical or the neck areas. And the lymph nodes are going to filter the fluid as it passes through. And in the lymph nodes, you do have a higher concentration of cells whose purpose is to carry out phagocytosis, so they can engulf anything that it recognizes as foreign. You're not supposed to be here. I'm going to eat you. And it also has a higher concentration of specialized cells, the T cells and B cells involved with the immune system. So the lymphatic capillaries, these are located where the interstitial fluid enters into the lymphatic system. They're near the capillary beds, near the arterioles and the venules. They are not found in the central nervous system, the bones, teeth, or cornea. There's little flaps at the end of them that will open in response to increased pressure, so that then will allow the fluid to flow from interstitial space into the vessel, thereby hoping to decrease the pressure, the swelling that you would be observing. Something else to keep in mind is that lipids and your fat-soluble vitamins also will enter here, especially in the location of the small intestines. And so this is showing over here, you've got your arteriole. Here it shows arrows obviously showing the flow of blood. And so some of the blood is going to come into this capillary bed here. And you have the filtration occurring. So things are moving out of the blood here. And as it goes to the capillary bed, as you return now, on this end of the capillary bed is where you have the reabsorption occurring. But as we said, not all the fluid gets reabsorbed, which is why you have the lymphatic, which are the green vessels here. So they are absorbing that excess fluid. And this is where you can see there are these little flaps here that will open. See how this one opened? And it's allowing the fluid to come in. Under low pressure, they would close. And then you have valves. Here's a valve right here. So once the fluid comes up here, that valve is going to close, preventing it from leaking back out again. So the lymphatic capillaries drain into lymphatic vessels, which are a little bit larger in diameter. They do have the valves to prevent the backflow. The large vessels are going to drain into even larger, what we call lymphatic trunks. And then the lymphatic trunks drain into lymphatic ducts. And then it's the lymphatic ducts that drain into the circulatory system. So it goes from the capillaries to the vessels, trunk, ducts, and then finally back into the circulatory system. In terms of the lymphatic ducts, you've got 2. You have the right lymphatic duct, which drains the right side of the body in terms of the right side of the head, the thorax, and the upper limb. It will drain. The right lymphatic duct drains into the right subclavian vein. And then the thoracic duct drains the majority of the body, and it will drain into the left subclavian vein. The cisterna chyli is a chamber. It's below the diaphragm. It receives lymph from the legs and the pelvis, and it's going to drain into the thoracic duct. So this picture here shows, I think it's really kind of a neat picture. Visually, you can definitely see that the right lymphatic duct drains everything that's in purple here. So it's not an even 50-50 the way it drains. And you can see in this enlarged diagram right here where you've got the right subclavian vein just before it merges with the right internal jugular vein. That is where, once again, green is the lymphatic system. That's where you've got the right lymphatic duct draining in. So it's draining all of this purple area here. And then the yellowish area is showing the area that is drained by the thoracic duct. So that whole left side, the lower part of the body below the diaphragm. In this enlarged area, this larger chamber right here, that is the cistern alkali. And then it's going to drain behind the heart and come in, and it also then drains into over here, the left subclavian vein. So the right subclavian vein, left subclavian vein. Well, we'll see in a bit where we'll talk more about the lymph nodes a little bit. Those sometimes when you have an infection will swell, and that is oftentimes in the neck. What physicians are, when they're palpitating your neck, they're trying to feel, you can tell the difference. You can tell if the lymph nodes are swelling. That's indicative of an infection. So what's triggering the infection? Well, it could be viral, it could be fungal, it could be bacterial. How does your body respond to infections? And this is where your immunity is going to play a role. There's 3 levels of defense. Some books will divide them, and in microbiology we tend to divide them initially into innate immunity and adaptive immunity. And the innate immunity is very nonspecific. So it's the way the body's going to respond, whether it's E. coli or whether it's pine tree pollen. It just responds. There's something foreign here. It's not part of you. We need to destroy it. And then the adaptive is very specific. So it's the way the body responds to something you've seen before. And so its response, it can distinguish between E. coli versus Staphylococcus aureus versus Streptococcus. It is very, very specific. Now, sometimes what we will do is we refer to these as 3 levels. In the innate, we're wondering where's the 3rd level. Well, the innate can be divided into 2 parts. There is the barrier, which is non-specific, and this is where some books will include this as a separate level or type, but a lot of people include it under innate because it is still non-specific. This is your first line of defense. It is a barrier, a physical and sometimes chemical barriers, like your skin. If your skin is intact, that is a physical barrier preventing microorganisms from being able to penetrate and invade into your body. It physically can't get through there. Now, if you have a cut, well, now you kind of opened a door to maybe allow things in. Mucous membranes, like what we see with the respiratory tract, what we see with the gastrointestinal tract. The mucus helps to prevent any penetration of pathogens. It's usually an immediate response. The innate, so you can have the physical barrier, and then you can also have cellular and chemical. It's kind of your second line. It's also very rapid, but once again, non-specific. So a pathogen was able to get past the skin. All right, you cut your leg and you had an opening there, and a bacteria was able to penetrate. Now what? Well, there's certain things that will happen in the body which are part of the second line of defense. And once again, it doesn't matter whether it's, say, E. coli or whether it's the fungus aspergillus. It doesn't matter. The response will be the same. Recognizes just that there's something foreign here. We have an injury. We need to clean up, and we need to prevent any further infection from progressing. It's kind of like when I teach it from the microbiology standpoint, I often say it's almost like a battle, like, oh, our outer, perimeter has been compromised. We need to send in more troops to fight this and don't allow them to get to the second line. And then the adaptive is going to be very specific. It involves the lymphocytes. It's going to be slower in its response, but it does have a memory aspect. So how's the immune system, how does it function? How does it work? Well, the cells are going to start as the hematopoietic stem cells in the bone marrow, and then they will differentiate. Some of the different types of cells, you have phagocytic cells. They ingest pathogens. They carry out phagocytosis. Lymphocytes, they will be involved with the adaptive immunity. That's going to be your B cells and your T cells. And then granulocytes, these are involved with the immune response as well. And so you start up here with the stem cells, and then some of them, not this fork in the road of this dichotomous key or flow chart, will develop into like eventually your monocytes, which when it leaves the circulatory system is known as a macrophage. This has phagocytic abilities. You're going to see where the neutrophils are also going to be involved with the inflammatory response, as well as basophils. The lymphoid stem cells, these are going to differentiate into a group that we call the natural killer cells, which carry out phagocytosis, and they can release certain chemicals that will kill cells, and then eventually end up with your B and your T lymphocytes. So those B cells we just saw, they will mature in the bone marrow. Now, both B and T cells initially develop in the bone marrow. The B cells stay there. They will mature there. Ultimately, they're involved with production of antibodies. Now, people will often confuse antigen versus antibody. Antigen is a marker that's on a foreign substance. So that's going to be on the bacteria, on the pollen, on anything that is not you. The antibody is something that you make in response to the antigen. Now, I know with blood typing we use the term antigen, and this is where I know it sometimes causes problems. When you're talking about immunity, the antigen is on the foreign substance. The antibody is what you make against the antigen. So the antigen is what you are seeing as, oh, that is not part of me. I need to attack and destroy. Who is making the antibodies in terms of what type of cells? Specifically, it's plasma cells, which are activated by the B cells. So it's when you see B cells, in your mind, be thinking antibody production. T cells, like I say, they initially develop in the bone marrow, but then they're going to move and they will mature in the thymus. There's several different types of T cells. There's helper T cells, regulatory T cells, cytotoxic T cells. So there's several different types, but all of them will mature in the thymus. They tend to destroy cells that have a pathogen in them, like a virus. They also are involved with sending chemical messengers, so they can activate other cells. The two types of cells or categories, the B cells and T cells, are directly involved with each other. They're directly related. With the T cells, that's often referred to as a cell-mediated because it's involving things at the cellular level, and the B cells are often referred to as the humoral immunity because it's involved with the antibodies. Plasma cell, that's a differentiated B cell that does secrete a specific antibody. And then you also have those natural killer cells I just talked about a bit ago. They will be involved with the innate immunity. They're very similar to cytotoxic T cells. They're definitely known for destroying cells that are infected with viruses. So your primary lymphoid organs are the bone marrow, because that's the site of where hematopoiesis, where you're producing these stem cells, and then that's also where the B cells mature. And then the thymus, That is a bilobe gland. It's between the sternum and the aorta. The T cells mature here. This is a gland that it starts out large at birth and it shrinks as you get older. And so, once again, this is showing right here where the location of the thymus, and then it's enlarged over here. Secondary lymphoid organs, these are sites with very high concentrations of B cells and T cells. May have both and may have one or the other. It also tends to have reticular fibers that will have macrophages that are fixed on them. Macrophages are capable of carrying out phagocytosis. Germinal sites are sites where you have very rapidly dividing and differentiating B cells. And then, oh, I'm sorry, you can probably hear I have a cat here who's wanting to play. And then you also, with secondary lymphoid organs, have very high endothelial venules. This allows cells from the blood to directly enter into the tissues. So the secondary lymphoid organs, these would include things like the lymph nodes, the spleen, the lymphoid nodules, which would include things such as the tonsils, the what we call MALT, mucosa-associated lymphoid tissue, and then the BALT, which is bronchus-associated lymphoid tissue. With the lymph nodes, their purpose is to remove and filter out any debris and pathogens from the lymph as it passes through there. So to be able to remove things like pathogens, it would make sense that it does contain both dendritic cells and macrophages, which carry out phagocytosis. Think of or visualize like little, if you want to think about a computer game, these little, okay, from the old days, Pac-Man, but you know, these little creatures, they're just in there and they're going to be chewing up, recognizing you're not part of me, okay, I'm going to eat you. You're part of me, you're free to go. It does also contain T cells and B cells, which, as we've been saying, are involved with the adaptive immune system. With the lymph nodes, all the lymphatic vessels that are draining into the lymph node are known as your afferent lymphatic vessels. So they're draining into the lymph node. The efferent are the ones that are draining the lymph out of the node, so as it's leaving. So afferent is arriving, efferent is leaving. In this picture of the lymph node, in the schematic drawing right here, one thing I want to point out, all of these are afferent. This will be afferent, this will be an afferent over here. And there's only two of these efferent. You have more flowing in than you have leaving, which makes sense if you stop and think about it. Because you're, well, wait a minute, isn't that going to slow it down? If you have, you know, if you think of, yeah, I've got all these entrances for the fluid to be flowing in, and as it's flowing through the inner area, you're going to have a roadblock, if you will, here. You're going to have a backup. It's like a big old traffic jam, if you will. There's only two ways out. But that makes sense because the purpose in the lymph node is to filter, is to remove. Maybe there's some damaged red blood cells that are going through there, and other tissue debris that's managed to flow through. Maybe you have these pathogens. Well, if you slow the flow down, that increases the amount of time that fluid is in here, which increases the amount of time. Let's say there's a bacteria. Then that bacteria may come in contact with a macrophage that then can engulf it. So you're increasing the amount of filtration. You are increasing the chances of cleaning up the lymph, which is the whole purpose of the lymph node. You also are increasing chances, once again, if there's, say, a bacteria or virus that's going through. If you have those B and T cells, then they have more chance of coming in contact and being exposed to. And a little bit later, we'll talk about what happens when it becomes exposed to something foreign so that then it can recognize it. So is it going to slow the flow up? Yes, it will. But is that a good thing? Yes, it is. Now, when you get an infection, you tend to have an increase then, excuse me, on the bacteria. It starts a response of fighting those bacteria, and oftentimes what will happen then is that the lymph node actually becomes even more swollen. And like I said earlier, you can feel when it's swollen. That gives you a clue. A nice, healthy individual should not have swollen lymph nodes. This is something else that they will do when they are treating for different types of cancer. It's depending on where the cancer is. They may check some of the lymph nodes nearby to see if there are cancerous cells in here. Now keep in mind that as the lymph is flowing through the lymphatic vessels and it passes through lymph nodes, it's not like there's just one lymph node that it passes through before the lymph goes into the blood. It's going to go through a series of lymph nodes. So the lymph may be collected from the capillaries, and it goes through lymph node number 1, then lymph node number 2, then number 3, then number 4, etc. They're in like a chain of them. And so you can check and see, let's say unfortunately someone does have cancer and they're checking the lymph nodes. Number 1, it would be indicative that, ooh, you don't want to see the cancer in the lymph nodes because that potentially can be spreading from where the original tumor was. It's no longer isolated to that area. But let's say that lymph node number 1 and number 2 do have cancerous cells in them. Typically, they would remove those lymph nodes. But then, let's say lymph node number 3 is clear. Then that means ideally, what you're hoping for is that if lymph node number 3 is clear of any cancerous cells, the cells did not get there, which is a good thing. The spleen contains macrophages and dendritic cells as well, once again to help remove any debris, remove any pathogens, and also to remove any of your erythrocytes that are dying. There's a lifespan, and when it gets to the end of their life, they start dying off. That's why you're constantly making more erythrocytes. And so what happens to the old ones? They're going to be removed, the spleen. The spleen is one of the organs that I find a lot of people are like, well, I've heard of it. I don't really know where it is. It's tucked over here on the left-hand side. In the picture over here, you can see here's your liver. In this drawing right here, you put the diaphragm down here. This is the spleen right here. You can see a kidney back here. And this is the pancreas, and your stomach would be right here in front of all this. And so here's this. It's tucked. Oftentimes it's kind of hidden by the stomach because it's kind of tucked to the side and behind the stomach area. Your lymphoid nodules are located in the respiratory and digestive tracts. Why in these areas? Because you have increased risk of exposure to pathogens from what you eat and what you inhale. The tonsils, excuse me, there are the pharyngeal tonsils known as the adenoids in the back of the throat, the palatine tonsils on either side of the throat, and the lingual tonsils that are at the base of the tongue. And this is showing the location, like here's the palatine, the lingual, as you can see from this diagram, and then way up here is the pharyngeal, and looking into the mouth here, your palatine. These are swollen. Somebody's got an infection. And then the malt and the balt. The malt is in the gastrointestinal tract. They're also found in the breast tissue, the lungs, eyes, and Peyer's patches, which are in the small intestines. And then the balt, which is in the bronchi, and also between the bronchi and arteries. So with your immune system, as I said, it's kind of a 3-step process. You have the innate, which is non-specific, which is your physical barriers, and then your internal defenses. Your first line of defense, that's skin, the mucous membranes. Let's keep anything from progressing further. or being able to penetrate actually into the body. Some of the chemical secretions, things such as your tears. Your tears contain an enzyme, lysozyme, that kills bacteria. Some of your digestive enzymes in your mouth as well will break down. If something is able to get past, excuse me, past this first physical barrier, then you've got your second line of defense. And once again, I often look at it from a microbiology standpoint, since that is my specialty area, and I often describe it as kind of like a battle. Bacteria, we often talk more so about bacteria, but it could be any microorganism, or who knows, maybe a parasitic worm, whatever it is that's not part of you. It's trying to get into you, and it's kind of like you're going, haha, I got this skin layer, you can't get past it because I'm being careful and I don't have cuts. And then if you think I also have mucous membranes, so you can't get into me because I've got this physical barrier. Well, then sometimes the organisms, they're like, huh, you think your skin's going to keep me out? I got a way around that. Some bacteria can break down parts of the skin, or if you get a cut, then they're in. So they have ways of trying to get around those physical barriers. So now they're into the body. Then your body has developed the second line of defense, these internal defenses. Once again, non-specific. It doesn't matter what the organism is. It's just that it's not part of you. Where you've developed these things such as the inflammatory response, which is going to attract even more phagocytic cells to that area to try to fight the infection. It's like, ooh, we have a breach in the wall. We need to come here and we need to seal it off and not allow the invader to get and progress even deeper into our system. And then your microorganisms especially have developed some ways of getting around this, and they can like, hey, you thought that was going to stop me? Huh. I'm in, and you're not kicking me out. And then that's when the adaptive immune system is going to come into play and say, we weren't sleeping. We saw you get past these two first and second line of defenses. We got a third trick up our sleeve. It takes a little bit longer to get it running, but we got you now. We have a very specific response now that we're going to use. So this is where sometimes you'll see this first defense, second, and then third line of defense. So that first, once again, it's going to be very rapid. It's non-specific. Second line, also non-specific. This involves macrophages, neutrophils, natural killer cells. It's going to be mostly things occurring at the cellular level. So the first line of defense, once again, skin is one of the biggest things. It's a barrier. The sweat may be annoying to you, but some of the secretions in the sweat, it actually kind of flushes things off the skin. It also has a slightly lower pH, it's slightly acidic. And you're like, well, what difference does that make? Bacteria prefer neutral to slightly basic. They prefer like 7.5, some even up to about pH of 8. Fungi tend to prefer a little bit more acidic. Now they prefer it more around 5.5 to 6. So your pH, just by lowering the pH of your skin, that simple thing is going to help prevent some bacterial infections. In your mouth, your salivary glands also contain lysozyme, same one that I mentioned in your tears, that are bactericidal, meaning they will kill bacteria. In your stomach is pH 2. It is very, very acidic. On your mucosal surfaces, you have this constant flow that's going to help prevent. You also, and this is a big, big thing, you have normal flora. This is something you may not want to think about, but your body is covered with bacteria. But that's not a bad thing. It's not a gross thing. Well, you might think it's gross, but it's not a bad thing. They're non-pathogenic. They're part of the normal bacteria that are there. They are supposed to be there. You do not want to wash them off. When you wash your hands, do you want to completely sterilize your hands? No, you don't. Because if you remove the normal, non-pathogenic, pathogenic meaning it causes disease, if you remove those, then it's like you're clearing the slate, allowing for an infection to start. It's kind of like if you think of a busy parking lot. If all the places are filled, there's no place for a new person to come in and park, is there? Well, if all the spots, all the niches are filled on your skin, there's no place for one of the bad bacteria to come in. And just FYI, of the known bacteria, only about 5% of them are known to cause diseases in humans. The other 95% are either beneficial to you or, no offense, they don't care about you. They're not pathogenic though. So just kind of keep that in mind. In terms of the cells of the innate immune system, you've got macrophages. These are going to be involved in both the innate and the adaptive immune system. Some of them are fixed on those reticular fibers that I mentioned in the lymph nodes. Some of them, they'll move very freely in the body. So they can move around kind of on surveillance, looking for any type of foreign substances. Neutrophils. These are going to be attracted to sites of where you have an infection or an injury by chemotaxis. There are certain chemicals released that will attract them there. They're going to be involved with the inflammatory response. Monocytes, these are precursors for the macrophages and the dendritic cells. Monocytes are in the blood. When it moves out of the blood, then it's called the macrophage. And then your natural killer cells. These are cells that they will induce apoptosis, which is known as programmed cell death. They will do this process where they will induce the cell death in your cells. So yes, your own cells are going to die, but they are your cells that are infected with bacteria or viruses. Think of it this way. And I'm just going to mention this now because I know a lot of times people say, okay, with viruses, why is it that people say that antibiotics don't work? It's very hard to develop an antibiotic to work against viruses because viruses spend most of their lifetime in your cells. How do you kill something that's in your cell without killing your cell that it's in? And that's tough to do. Bacteria are a little bit easier to treat because they don't necessarily spend their whole time inside your cell. A lot of them never enter your cell. And bacteria are prokaryotic cells. They are similar to our cells, but there are also some major differences because your cells are eukaryotes. And so we look at what those differences are, and that's one thing that we really focus on when developing antibiotics, are the differences between them. Fungal infections are hard to treat because fungi are eukaryotes. Their cells are even more similar to ours. They don't have the differences like the bacteria do. So that's, once again, how do you basically kill something that's similar to you, very similar to you, but how do you do that? It's tough to do. And like I said, the viruses, one of the big problems is that they spend most of their lifetime in your cell. How do you know they're there? It's kind of like if you were to drive down a street, just pick a random street, drive down, look at the houses. Which houses do the owners have cats? Can you tell by driving down the street? You might not be able to tell that. And so it's very tough. There might be some little signs, and that's what the natural killer cells will do. Typically, when viruses infect a cell, there's a little signal that's put on the surface of the cell, and that's what the natural killer cells are going to look for. So how are your cells able to recognize the pathogens as being something different from you? Well, the macrophages, the neutrophils, the monocytes, and the natural killer cells, they have special receptors. They're known as the pattern recognition receptor, the PRR. So those are on the cell at all times. It can recognize a huge wide variety of pathogens. It can recognize damaged cells at all times. Sometimes this is referred to also as a self versus non-self, that they're able to recognize things. What are some of the other aspects of the innate immunity? Well, there's some soluble mediators, things like cytokines. These are signal molecules. So one cell will secrete it, and then it will travel through the interstitial fluid, bind to a second cell, and the second cell changes its physiology. Chemokines are chemicals that will attract cells by chemotaxis. Early induced proteins, these will be made only when they're needed, such as interferons, and then complement. This is a series of proteins that are going to become activated. Usually a couple of the proteins that they become activated, it's what we call a complement cascade. The first protein activates the second protein, which activates a third one, and might have a third one come in, another one come in and bind, and it's this whole series of steps. What's the end of result of the complement? Death of the pathogen. And it's also going to be attracting more phagocytic cells to the inflammation site. And so this is just showing in this diagram here of you have your pathogen, say your bacteria here. It's got the antigen. Remember, that's what's on the surface of the pathogen. Well, what's going to happen over here, this blue thing here, this is the antibody. So this is on your cell. It starts triggering, as you can see, the C1 triggers the C3 to C4, and then the C3. They're not numbered sequentially because they're numbered in the order they were discovered. But you can see, in order for, say, this step with C5 to happen, this step has to happen, which has to have this beforehand, which has to have this beforehand. So that's what we call a cascade. Everything is dependent on the previous step. And what is the end result of a complement? The end result is that you form this complex, which is basically it's like someone going in on if you have a water balloon, and somebody comes in and they start poking with a needle or a pen, a knife, if you will, just something sharp, and you start poking it. What's going to happen? Well, the water balloon, you're going to get wet because the water balloon's going to pop and the water starts leaking out. And so the end result of complement is that you end up destroying this pathogenic cell. It's poking holes in the membrane so that all the inside starts to leak out. Well, that's going to kill the cell. So the ultimate thing is that cell death will occur. So I've mentioned the inflammatory response a few times. So what is the inflammatory response? You have four classic characteristics: heat, redness, pain, and swelling. And sometimes you have loss of function, temporary loss of function. There's tissue damage that has occurred. You cut yourself, whatever that initial injury was. What's going to happen is that mast cells come into the site and they're going to release histamine. Histamine helps to initiate the inflammatory response. It's releasing, the histamine releases chemicals that attract phagocytic cells. Why are they attracted? Because of the chemicals. Chemotaxis is the attraction. You can have positive or negative chemotaxis. Negative is where chemicals released and things want to go the other way. Positive chemotaxis is where they are attracted to it instead of being repelling from it. Vasodilation. Your blood vessels near the area of where the injury is will dilate. That increases the blood flow to the injured site. That is one thing that helps to attribute to the redness to the area. You also have increased temperature because blood has heat with it, so that plays a role with the warmth or the heat to the area. The blood vessels have increased vascular permeability, which means it's easier for things to pass out of the blood into the interstitial fluid, so you have an increase in interstitial fluid. That's why you have swelling in the area, and then all that increased swelling is pushing up against the nerve endings, and that's going to trigger pain. Why are you doing this? Because the body has recognized that there is an injury. You need to trigger that inflammatory response. Why? To attract the phagocytes to the area, because there could be foreign invaders, if you will. There could be bacteria, there could be viruses, there could be fungi that have been able to now penetrate. It's like we've had a breach of the outer perimeter. We need to seal off this area. You want to prevent a potential foreign pathogen from invading even deeper into the body. So you want to contain any breach to that area. Increase the blood flow to bring in the fighters, bring in the phagocytes that recognize and attack and destroy any foreign invaders. You also need to clean up. You've had an injury. You're going to have tissue damage. You're going to have debris there. You need to clean that up. And the phagocytes will also clean up any damaged tissue that's in the area. In terms of phagocytes, neutrophils will be the first ones to arrive, and then the macrophages will arrive second. And so this picture here is showing you've had a cut. So you're showing here with the epidermis, you've got a cut to the site. You've got various bacteria that have been able to penetrate here. You do not want them getting here to the blood. You want to try to contain them in this area. Don't let them go any further. The damaged tissue releases certain chemicals that act as positive chemotaxis, attracting some of these are mast cells that are releasing histamine also, which then are going to attract the phagocytes to come to the area. What do? They're going to basically eat up these guys. You have increased permeability, so you're increasing the fluid here. You're starting to get some swelling around here as compared to the initial site. That, as I said, is going to push against the nerve endings all throughout here. So it's going to start hurting, besides the initial hurt of the injury itself. You have, because of that increased flow to the area, you're going to have redness, warmth, and that swelling. You want to prevent those bacteria from getting into the blood supply. Well, that can work, but at the same time now, we're going to also want to activate the adaptive immune system. This is a very specific response. This involves the antigens on the surface of those foreign cells, and you're going to want to recognize those specific antigens. Who's going to recognize them? The B cells and the T cells. Now, sometimes we talk about the primary adaptive response and the secondary adaptive response. The primary adaptive response is when the very first time you've ever seen that pathogen, so it's your first exposure to it. Oftentimes here you will actually have the disease occurring. The disease is usually more severe. Just with some of the terminology, we don't always talk about it in A&P, but in microbiology, just so you're aware, there's a difference between infection and disease. Infection just means that the pathogen is in you or on you. Disease is when you've had a change in the normal physiology. So you have the manifestation of that infection progressing and having adverse effects. So physiologically, your body's not acting under normal and doing the normal things that it should. And you see results of this. You may be tired, you may have upset stomach, you may have shortness of breath. And so we make that distinction. You can have an infection and not have disease, meaning you could have the pathogen in you, but you never know it because you never get symptoms of the disease. Primary response, that first exposure, usually you're going to be sick because you've never seen it before. It takes a little while, remember, for the adaptive immune response to kind of get up and working. It's got to be exposed to it. It needs to start up, as you're going to see, a whole series of steps on how it's going to react to it. The secondary adaptive response is when you have any subsequent exposure. So not the first time, it's the secondary and any other times you're exposed to that pathogen. One thing with the adaptive immune system is that it has some memory cells. And so the response on the secondary time will be much stronger and much faster. It's like, oh, we've seen you before, we're going out full press here, and we are not going to allow you to cause any damage. And that's when you have the infection but may not even know it. The adaptive immune system is able to distinguish between self versus non-self. What's you and what is not you. So it involves T cells and B cells, as we have said. The antigen is a substance that's on the surface of the pathogen. It's oftentimes either a protein or a carbohydrate or a combination of both. The antigenic determinant, often referred to as an epitope, is just a small fragment of the antigen where a receptor can bind to. And we're going to talk about the T cells and the B cells in a moment in detail. One thing to keep in mind is that T cells will control and regulate the B cell response. So when we look at, these are showing various T cells. And the antigenic determinant, so this is a whole protein that may be found on, let's say, a virus or a bacteria. And it's just a piece. So the antigenic determinant is not the whole foreign thing, it's just a part of the foreign substance. The major histocompatibility complex, known as MHC, that is going to assist with showing the antigen to the T cells and B cells. There's two different types. There's MHC class 1 and then a class 2. Most cells have the class 1. It helps to stimulate the cytotoxic T cell response, which is going to result in the death of the pathogen. The class 2 is found only on the macrophages, the dendritic cells, and the B cells. So you have cells that are known as antigen-presenting cells, your macrophages and your dendritic cells. What do? They see a pathogen. They go, oh, this is not part of me. So they're going to engulf it, and they're going to destroy it, chew it up, and eat it. They're going to break down the cell. Pretty good at recycling, use what they can. Well, one thing that they will do, those antigen fragments, that is going to bind internally with the MHC. So inside the cell, the MHC is going to bind to this antigenic fragment. Not the whole thing, just the fragment. And then that complex, that antigenic fragment with the MHC, it's going to move internally, so it's going to move to the surface. So now it's exposed. And it's kind of like, okay, I ate up this foreign thing, it wasn't part of me, well, I'm going to take part of it. And when it binds and makes that complex with the MHC and then shows it on the surface of the cell, it's kind of like, that's why we call it antigen presenting cells. It's presenting it to the T cells. It's like waving a neon sign or a big old flag going, here it is, guys. I got something here for you. So it's not the antigen on the pathogen surface that activates the T cells. The T cell has to be activated by an antigen presenting cell. And this picture I know looks rather complex, but we'll kind of go through. So here, down here is a pathogen. bacteria, let's say. Does the markers, does the antigen on the surface of the pathogen activate this T cell? No. Now this T cell is just kind of floating around. Has it been activated yet? Not yet. Here is a phagocytic cell. So it's going to engulf the pathogen. It's chewing it up and eating it up. And what does it do? These structures right here are your MHC proteins internally. And now this is where it takes a piece of the antigen and it puts it on the MHC. And now it's going to put this on the surface of its cell. So here it's waving the flag or turning on the neon light, however you want to think of it. And it's showing this is what now activates the T cell. It's like this little yellow oblong shaped structure right here. That's what you need to be on the lookout for. That's what's on this guy. He's here. Go look for him. This is what you're looking for. Go get it. And so this is showing the process of where you have, how do you make the T cells? Because there's different types of them. So you start off with your thymocyte. It's going to go through a process here where there are certain markers on here. Can it bind? Yes or no. If it doesn't, there's a whole like weeding out process. So in the thymus, where this, remember this initially is in the bone marrow, then it moves to the thymus. So there's this whole process of can you handle the MHC and not recognize your own cells? You don't want a T cell to recognize your own cells as being something foreign. So it can't pass the first test, whoop, you're out, you're done, you're dead. Those that survive that continue on, and now it's going to go through additional selection, still in the thymus. If you're producing cells that are going to be functioning and recognizing and distinguishing between your cells and foreign cells, you better make sure they don't recognize your cells. Is essential. So there's this whole selection process. And then finally, what happens is eventually you end up with what are known as your CD4 and your CD8 cells. The CD4 are going to, down here you can, if you enlarge it, you can see a little bit better. It says CD4 positive, CD8 negative. So a CD4 cell becomes a helper T cell. The CD8 cells will become what's known as your cytotoxic T cells. And when cells become activated, once these become activated to a specific antigen, so once an antigen presenting cell comes in and shows itself and activates these cells, the helper T cells are going to turn around and activate more cytotoxic T cells. These are going to go out and be able to recognize that antigen and binds to those pathogens and kill them. That's why they're called cytotoxic. The helper T cells are going with the CD4 marker on them. Those are going, so they help to initiate and activate more cytotoxic T cells. They also play a role in activating the B cells. So those CD4 cells are very important in helping after the antigen presenting cell shows this, hey, here's the new markers, be on the lookout for this. The helper T cells are very important in helping to stimulate the other T cells and B cells for the adaptive immune response. And why am I focusing on this so much? Is because here's something of a practical application for it. The HIV virus, one of the things that it does is it wipes out the CD4 cells. Bad for you, good for the virus. If you wipe out the main cells whose job is to activate your immune system from a pathogen standpoint, that's a pretty clever thing. You just wiped out the surveillance. You took it out. It cannot see you now. It cannot activate the cells that can recognize a cell that has the virus in it. Why? Because you just took it out. You took out the whole security system. Like I said, that's pretty clever from the virus standpoint. From your standpoint, you just got your immune system wiped out. So now, not only are you susceptible to the virus that wiped it out, you are susceptible to any foreign invasion, which is going to overwhelm your body, the systems, as it's trying to stay healthy. But the thing that's supposed to help you monitor and prevent infections, that's been wiped out. Not a good thing to happen. In the primary adaptive immune response, both the effector T cells are produced, those that are working right now, the cytotoxic T cells, the B cells, etc. But also memory T cells are going to be produced. The memory T cells are long-lived, and that's what's going to be there for any subsequent exposure that allows for a very rapid response. Like I said earlier, the response is going to be immediate. You don't even realize that you are infected. The T cells, the different types of them. You have helper T cells. As I said earlier, those have a CD4 molecule. They do secrete cytokines. They help to stimulate other components of the immune system. There's actually two types of helper cells. There's helper 1 and helper 2. The helper 1 cells tend to target the macrophages, go after extracellular pathogens. And then the helper 2 cells stimulate the B cells. Like I said, it's going to activate them. And the B cells will then differentiate into plasma cells to produce antibodies, once again go after extracellular pathogens. The cytotoxic T cells, they tend to target infected cells that have an intracellular pathogen in them, like viruses. And then you also have regulatory T cells. Sometimes these are called suppressor T cells. They regulate the helper and the cytotoxic T cells. You need to have these in correct proportions. You don't want either one of these guys getting out of hand. If helper T cells get a little over anxious, then you can end up having an extreme response when maybe you don't really need it that bad. Or the cytotoxic T cells. Once again, you need to have a checks and balances, and that's going to be what the regulatory T cells do. So the T cells are often referred to as cell-mediated immunity, and the B cells are referred to as humoral. They are involved with production of antibodies, but the activation of them is dependent upon those helper T cells, which have that CD4. That is one of the reasons why, as I just mentioned, the relationship of how the HIV virus works with the inhibiting, basically, the helper T cells to monitor the progression of the disease. AIDS is the disease that you see manifested in late stages of the HIV virus. So you can have HIV virus and not have AIDS. And one of the things they do is they monitor the levels, and they'll often just record it as CD4. And that's what they're talking about, is what is the concentration of these helper T cells. So with the B cells, what's going to happen here is you can have the antigen on the bacteria, once again, can activate. And the helper T cells will activate the B cells that have been exposed. And ultimately, what's going to happen is the activated B cells now, some of them become memory. So once again, subsequent exposures, you can start having a response immediately instead of having a delayed response. And then a lot of the cells right away start to produce these antibodies. What do these antibodies do? Well, they're now in the bloodstream and they can float around anytime they see a bacteria with that specific antigen. They will bind to it, and that's marking them. And then you have further reactions that can attract cytotoxic T cells, that can attract the macrophages, when it can cause clumping. When you have the antibody-antigen complex, it's like, once again, flagging. Hey, come get this. We found the invader. Come get him now. So what do the antibodies look like that are being produced? They're glycoproteins. They contain heavy chains and light chains. So there's 2 heavy chains and 2 light chains. There's a constant region, a variable region, and an antigen binding site. So the heavy chains are these 2 chains that are on the interior. They're longer. Think of it that way. Yeah, they're long, so they're going to be heavier. The outer chains right here, the green and then the yellow, are the light chains. On the end, if it looks like a Y, like the ends of the Y up here, those are the variable regions. And then the constant region is down here. The stem down here as well as this area. So these will be the same between all of them. The variable region is a different region. And once again, if you think of this as the Y, on the tip of the Y, that is the antigen binding site. So that's where it's going to physically bind to that antigen if it comes in contact with it. There are different classes of antibodies. There's 5 of them. Some of the shapes are a little bit different. They have that basic structure that you just saw. An IgM is a pentamer, so it means you're going to have 5 of those connected together. IgMs are produced in the primary response, and they will activate complement. Remember, complement ultimately is going to end up with the death of the pathogen, so that's a good thing. So IgM is with the primary initial response, and then you're going to see a few days later IgG. IgG is a monomer, so it's only one of those forms of the antibodies. You'll see a picture in a moment. IgG is a little bit later in the primary response. So initially you have IgM, and then a few days later you're going to see the IgG. And IgG is going to be the huge with the secondary response. So any subsequent exposure, you'll see the IgG. IgG can also cross the placenta, which means some of those antibodies from the mother can cross the placenta and provide some protection for the developing fetus. IgA is a dimer. That means you have 2 of them. The stems connect together. IgA is found in mucus and tears, saliva. It's found in colostrum in that breast milk, so that's why it's able to provide protection for the newborn if the mother is breastfeeding. IgE is a monomer. This is prevalent with allergies and parasites. And then IgD is also a monomer. It's involved with the B cell receptors. This, for a long time until just recently, we really didn't know what the IgD exactly did. So research is still going on to determine exactly how it's reacting with B cell receptors. And so this is just giving a summary, but it's also showing the pictures. So like the IgM, the first ones to appear, like I said, is a pentamer. Here's the IgG, which is most numerous and is evolved late primary and then also the secondary responses. And then IgA up here, like I said, it's a dimer, so they're connected right here. Clonal selection of the B cells. Once you have an antigen has activated the B cells, you've got activation here. You're going to have your memory cells over here. So these will float around, be ready anytime in the future. As soon as it sees, it'll start production of additional antibodies. Meanwhile, for right now, these cells, what happens is the activated B cells stimulates the plasma cells, which stimulate, so up here it will stimulate the production of the antibodies. So antibodies are made by plasma cells. Plasma cells were activated by the B cells. So when you have an exposure, the initial exposure to whatever the pathogen is, you're going to initially see your IgMs and you see your IgGs. This is your primary response. You're fighting the infection. So during this time period, on the initial response, you're probably not feeling really good. And then you recover. So now you're feeling better. The level of antibodies go down, but you have those memory cells that are there. You're exposed a second time, and you don't have this delay. You have a much stronger, much faster response. Like I said, you may not even know that you're sick. When people talk about titers, a lot of you are going into the healthcare field, and either before going into your programs or at some point maybe ask, well, what is your titer for fill in the blank? What they're talking about, titer is the concentration of antibodies in your blood. That gives you an idea of what are those memory cells that are floating around. Why is that important? You want it to be high enough, let's say titer level for measles. If you've had the measles vaccine, you want to know that those titer levels are high enough, that the concentration of antibody is high enough so that if you are exposed, boom, you're going to get a good response and not get sick. If the titer level's too low, and it is different depending on whether you're talking like measles versus hepatitis B, whatever, they have certain levels depending on which you're talking about. You may need to get a booster vaccine too. What that would do is then stimulate to get those antibodies up to higher level, because you don't want them down here at 0. You do want them up. With immunity, we talk about active immunity versus passive immunity. Active immunity is when a person has been exposed to the pathogen or the antigen. You may not be exposed to the entire pathogen. You may just be exposed to the antigen. And that has triggered an antibody response in you. This confers long-term protection for you because you're going to have the antibodies. You're going to make those memory cells that are there. So if you have any other exposure in the future, you'll have an immediate response. Passive immunity is when an individual has not been exposed to the pathogen or the antigen. Instead, they've received antibodies. Did they make the antibodies? No. They received antibodies that somebody else made. Now, this will give you short-term protection. It will protect you right now, but not a year from now. Why? Because you didn't make the antibodies. If you didn't make the antibodies, that means there's no memory cell production. But it helps to protect you right now. Now we put these together. So remember, active is when you've had it. Passive is when you're receiving just the antibodies. So we have 4 categories, if you will. You have passive, naturally acquired immunity. So this is when an individual has received antibodies, because it's passive, naturally acquired. They got those antibodies in a natural means. Either the developing fetus was protected because the antibodies the mother made past the placenta, or a newborn who is breastfeeding, the antibodies are being passed through the breast milk and protecting the infant. Will that provide protection for the infant right now? Yes. Long term? No, because they don't have the memory cells. Passive, artificially acquired. Now the immunity is gained because, yes, you received antibodies made by somebody else, sometimes made in another animal, and they are injected into you. Sometimes they will do this when someone is sick or you're trying to prevent them from getting sick, but maybe they're immunocompromised and they cannot take a chance of being exposed to the actual pathogen. This was used as a means of treating individuals several years ago when there was a huge Ebola outbreak, and there was an American physician who went over to Africa to help out with the outbreak. He ended up contracting Ebola and survived. When they tested his blood, he had a lot of, obviously, of antibodies that he had made against the Ebola virus. Shortly thereafter, there was a nurse who also then became sick. And what they did is they were able to take blood sample from him, separate out just the antibodies, and then give the antibodies to the nurse to help her in fighting the infection. And it worked. This doesn't always work. It takes a lot of research to try to figure out when it will work, when it won't. One of the best ways to confer immunity is by active, because you will produce your own antibodies and you produce the memory cells. So you naturally acquired, meaning you were actually exposed to the pathogen. You got measles, you got chickenpox, you got whatever, and the flu, and you built up your own immunity. You've got those memory cells now. The other is artificially acquired active. So you're going to make your own antibodies. How are you exposed to it? Through vaccine. Now, many years ago, when they first started with some vaccination, they would give you actually the full pathogen. What we have learned is, and you've just learned the past bit listening, that you don't have to have the whole pathogen to trigger the immune response. You just need to have that antigen, the antigenic determinant. So, as they were trying to develop all these different vaccines, years ago, they would try to use the whole pathogen, and then they realized we don't need to do this and take a risk of someone developing the disease anyway. If you take just a portion, if you just take that antigen protein marker, and proteins work very well as far as triggering an antibody response, you just take a portion of the markers. If you're taking just a portion, there is no way that you can get sick because you're not getting the whole pathogen. There's no way possible. But you can still elicit a very good immune response and give you the protection so that you never end up with the disease. Now, some of us are old enough. I remember in elementary school receiving the smallpox vaccine. When I was growing up, everybody got that. Measles, mumps, rubella, which is German measles, polio. A lot of people, for a while, the vaccination program has been very, very effective in providing immunity and protecting, especially in what used to be called the childhood diseases, protecting children from getting the diseases. Some of these have very devastating, horrible effects if you get and contract the disease. The vaccination program has been very, very effective. Some people think, oh, well, measles doesn't exist anymore. Polio doesn't exist anymore. The cases are really low because the vaccines have been so effective. But the problem is, the only disease that has been completely eradicated globally is smallpox, and that was done about 78, 79. They had the last case. We have decreased the numbers of measles until recently. We've had some major outbreaks. We've decreased the numbers of polio. Polio has not been eradicated yet. The problem is when some people start thinking that these have been eradicated or such low levels, now I don't have to worry about dealing with vaccinations anymore, and so they don't do it. Now you're getting a whole population of people who are not protected. And that is a huge, huge chance that people are taking with their children, especially because a lot of these are given as childhood vaccinations. And that is a huge, huge gamble that you are taking. There's no reason why anyone, especially a child, should be developing polio in this day and age. I am aware of the fact that in the 80s, I believe it was early 90s, there was a report that was published, and a lot of people jumped on it, where they felt that there were side effects from the vaccines that were detrimental, that could potentially lead to autism. No one was able to repeat those results. And finally, a few years ago, the person who wrote the initial paper came out and admitted that it was fraud. He lied. He doctored the data and he lied about it. Unfortunately, that has not made the headlines like his initial paper did. It's about protecting everyone, especially the children. Immunodeficiencies. This is great when the immune system works the way it's supposed to, but unfortunately, sometimes things don't work the way they're supposed to. Sometimes people may be born with an inherited or a genetic disease that makes them more susceptible to opportunistic pathogens. Opportunistic pathogens are ones that, in a healthy individual, where the immune system is working properly, it's not going to cause you any problems. But, you know, if your immune system is not working 100%, they see the opportunity, that door open, and they're coming in. As I mentioned, HIV, human immunodeficiency virus, that interferes with those CD4 cells, those helper T cells, and it wipes out your immune system. They do not have a cure. They do have drugs, a combination of drugs, so they call it a cocktail. It's about 3 different drugs that they use that helps to boost the immune system, helps to boost up those levels of CD4 cells, and therefore it delays the onset of AIDS. But there is no cure. They have been trying to work on a vaccine. One of the problems with viruses, especially the HIV virus, this class of viruses, which is in a similar class, RNA viruses, just like coronavirus, etc., is they tend to, if it's an RNA virus, they tend to mutate more frequently. And that obviously causes a problem when you're trying to develop a vaccine, and the virus keeps mutating on you. So now it's not the way it was when you first started working with it. There's also a huge issue in terms of ethics on how do you test vaccines. It's one thing for coming up with new vaccines and wanting to test them when it's something against the flu or something else that may make you sick, but it's not going to kill you. Ethically, you cannot test on humans a vaccine for HIV when you know there's no cure for it. You cannot do that. And so it's a very slow process. Yes, you can work with animals, but it's a very slow process in trying to develop this. Hypersensitivities. Once again, sometimes that immune system is working a little bit too good. There's 4 different types. Type 1, this is where you have an immediate reaction. This is where your allergies are going to play a role. It's really a case of your immune system is just a little too good. For me personally, I'm allergic to tumbleweeds. Not a problem in Texas, big problem when I lived in New Mexico. Sometimes the pollen from things, your body's seeing that, oh, this is something foreign, let's attack this. Well, is the cottonwood pollen going to kill you? No, but your body's response, you think it is. It's just overactive. Type 2 hypersensitivities, it's going to involve complement, where it's wanting to destroy things. An example of this is when, say, blood transfusion is done and you've given the wrong kind. It recognizes it, starts to destroy it. Type 3 hypersensitivities, these are things like glomerulonephritis. This is a disease affecting the kidneys. Rheumatoid arthritis, it's attacking the joints. Systemic lupus, all these, those are types of autoimmune disease where your immune system now is recognizing some of your cells as being foreign. Type 4 is delayed. So type 1 is immediate, type 4 is delayed. So you don't see anything initially, but maybe 24 hours later, 48 hours later, now you start to see a response. If any of you have had a TB test, that is an example of a delayed response. That's why they give you, to do a TB test, they give you the injection, and then you have to come back in 24 or 48 hours, and they check to see if there's reddening at the site. Contact dermatitis. Sometimes examples of these will happen where, once again, you touch something that you have a sensitivity to. Some people have a metal sensitivity, so like jewelry, they may have to be careful of the type of metal it's made from. Poison ivy. Do you break out in a rash of poison ivy as soon as you touch it? Not usually. It's delayed. Autoimmune responses. Basically, what's happening, as the name implies, is the individual's immune system can no longer distinguish between self versus non-self. It attacks everything as being foreign, including your own cells. Usually the treatments are treating the symptoms and are also going to be immune suppressive. So if you're taking an immunosuppressive medication to keep the autoimmune disease under control, that means you are at a higher risk for any other type of infections. Lupus is an example of this. As an example, I have a family member a sister-in-law who has lupus, and so she, because of the medication she's on, she knows that it's immunosuppressive, and so that puts her at a much higher risk for developing any other type of infection. Because of this, she has to be very careful. She tries not to be around anyone who is sick during the COVID-19 outbreak. She basically had to quit work, as many people did, and she also, because my nephew, who worked at a grocery store, and my brother, who worked in construction, were considered essential workers, so they had to continue working. She had to pretty much isolate in her room and could not see them. She just couldn't take the risk. Which, I mean, that's tough for anyone under any circumstances, but we all knew with COVID-19 initially that if she were exposed to it, we knew basically it would kill her. So she went for, just because of circumstances, the only way she could talk to her 18-year-old son, who was a senior in high school, for over 2 months. She could not physically see him. She talked to him through between a door. So it's a tough thing to have to deal with. In terms of transplantation and cancer immunology, anytime they do a transplant, they're going to do tissue typing. You are trying to match those compatibility complexes as closely as possible. Some parts of it obviously are unique, so each individual is different, but if you can get it as closely matched as possible, then you're going to reduce chances of rejection. There's still a chance for rejection, but you just want to put the odds in your favor. The recipient is going to have to be on anti-rejection medication for the rest of their life, which is going to reduce their immune system. It's going to suppress it. The odds are, however, or the chances, if you're having to receive a transplant, you've run out of other choices. In terms of cancer, they often refer to elimination, equilibrium, and escape. Elimination, your immune response is killing most of cancer cells. There are certain markers usually placed on cancer cells where the immune system can recognize some of these aren't quite right, let me kill them. Equilibrium is when any remaining cancer cells are kind of held in check, so you don't have the development of a tumor. If some of those cells mutate, they're able to evade the immune response, so they're able to escape that, then you have the development of the disease. A lot of cancer research and newer treatments, or trying to look at monoclonal antibody type of research, where basically you are developing treatments that are specific for you and geared just for you. So it's referred to as the lymphatic system, and it works with the immune system. Both are working for protecting you. It's your defense mechanism.